Okay, so for the sake of those who have joined just a few minutes after I started, I think I should just start from the beginning so that you hear everything that I started to talk about. I welcome you all to SBT 214, Biomembranes and Enzymology. As the course title suggests, this course is going to be divided into two main parts, which is biomembranes and enzymology. We'll start with enzymology. We will go through some fundamentals of enzymes, and then we'll continue to look at biomembranes. So these are the two major parts in which the course is divided. We've met before in first year, second semester, so we are acquainted, and so there will be no need for me to introduce myself. So as I was saying earlier, even though the program which you have been given is that for the first 3 weeks of the semester you will be online, and then after that you start to have some face-to-face lectures. But for this particular course, we are going to be almost throughout the semester going to be online, and that is because I am currently not in Ghana. So we are going to be having our lectures. Almost all the lectures are going to be online. Hopefully, getting to the end of the semester in July, if I happen to be in Ghana, then we can have some face-to-face lectures before you get into your exams. So as I was saying, the course divided into two major parts, biomembranes and enzymology. What we'll do is take a look at the naming and classification of enzymes from the beginning. And then we'll continue to take a look at the roles, the functions of what we refer to as coenzymes and cofactors. We'll continue to take a look at mechanisms of action of enzymes. And then we look at kinetics. There we'll look at some processes of inhibition of enzyme activity. As we go through, we will from time to time look at some practical applications of these things in industry, different industries, maybe food, beverage, pharmaceuticals, or say medical field. We will take a look at the applications of the knowledge that we are getting in these fields. And then when we get to biomembranes, which is the second part, first of all, we want to understand the structure of fatty acids, triacylglycerols, just to say generally lipids. I don't know if you did study lipids in first year or even second year, first semester. I don't know if you did anything on that. You can actually write in the chat if anything, if you want to respond to what I'm saying. If okay, so Esther says yes, so you did some things on that, which is good. We'll go over them so that you can remind yourself over these things. Perhaps there will be certain things that you didn't cover. It's important that you understand that very well before we move on and start to look at the structure of lipids, and then the structure of biomembranes. And then we can look at also some processes of transportation across the membranes, and also look at what we refer to as receptors on the membranes, which are responsible for what we refer to as cellular transduction or cell communication. We'll take a look at those processes. In transportation, we'll look at different forms of transportation. And then when we take a look at the structure of the biomembranes too, we take a look at things like asymmetry of the biomembranes, the types of movements of the lipids, or what we refer to as membrane motion. We'll take a look at all of these things. Then, of course, when it comes to assessment, okay, so this one, I think you should pay attention, because a lot of times people keep asking the same questions over and over again. So now, as we are having our lectures online, what I will do with you, I mean, this is what I did with those that I taught in first semester. I'll give you self-assessment questions from time to time to cover for everything that we are going to do. You have self-assessment questions, and these self-assessment questions, they are for you. You are going to use it. You solve those questions referring to the materials I'll give to you. That is the lecture presentations. I'll give you some books. You make use of these materials together with the lectures as we are having being recorded. The video will be available every time when we finish the lecture. I will share the link with a class rep, and a class rep will give this link to you. Of course, the link usually on the Microsoft Teams is active for, I think, 90 days, 3 months. After that, the link expires and the video goes into archive. So I will actually upload the video also onto my channel and share that link with you, so that one is there all the time, so you can always have access to it. And then once you make use of these materials, you answer these questions. It's not that you answer them for me to mark, but it is for you to understand, because I will give the questions particularly on the same things which we are doing at the lectures, so that by the time you finish listening, you finish listening to the lectures and answering those questions, finding answers to them, you will have a better understanding, a deeper understanding of these things. Now here, I will say that this is to leave more responsibility to you, because at this point in the university, as young adults, you should be having more and more opportunity to take responsibility of your studies and everything, actually, because from now on you're going to translate into being adults in society. So you have to get opportunity to be responsible. I say this because last semester, when I gave these self-assessment questions to students to go use the materials I give them to find answers to questions and all of that, instead of some people, instead of them sitting down and making good use of all these things, lazily just go and type some prompt in some generative AI and produce some answers with garbage, and they keep that garbage in their minds and produce in exam. If you do that, you have yourself to blame. So this is where the responsibility comes in. If you have all the materials that are given to you to help you to answer questions that are given to you, make use of that. and not lazily just go wanting to generate any garbage from somewhere to say that is what you are going to now keep as answers. Now, this is also important because in exams, it is just out of those same self-assessment questions that I'm going to select some of those self-assessment questions and give you as exams. So it means that your participation or your engagement with the self-assessment questions which I give you, the lectures that we have, the videos, the materials I give to you to use to study, your engagement with all of these things is going to reflect in the exam. Because if you really sat down and solved the self-assessment questions, then you shouldn't have any problem in exams. But if you didn't, that is where you are going to have problems. So I'll urge all of you, sit down, solve the questions when I give them to you. Make use of what I give to you, the materials. Of course, if you have some other books, biochemistry books, there are so many of them out there. So if you have any of them which you could use to, solve those questions, no problem. Just don't go and generate any garbage, because of course, it's not everything which generative AI is giving you which is accurate. So if you don't pay attention to use the real material that I give to you, you're going to have problems. Now, so that's about exams. How about the continuous assessment? So we're going to have lectures online almost all the time. Again, this is for you to be, responsible. And also, I consider the fact that, yes, it's possible that on some occasions, one or two people may not be able to be live at lectures. But of course, you must also know that as we are recording the lectures, right after the lecture, automatically attendance is generated, which is actually very nice, interesting, because with your numbers in the class like this, if we were meeting face to face, it will be difficult to be marking register for you every time. But this time it is even easy. So automatically you have the register marked. Now, even if it happens that one or two people are not able to attend a lecture or two, the videos are still there. So that's the advantage of this online platform, that the video is always there. If it was face to face, the video will not be there. And so if you don't get it face to face, it becomes difficult to now sit down trying hard to remember. But this one, the video is there, so you can watch it several times, as many times as you want, over and over again. If you don't understand anything, you can repeat it. You can even slow down the video and hear it much, much clearer. So it's an advantage of this system. And so if one or two people, you are not able to attend, the video is still there. So still, you still have the opportunity to engage with the material which is given to you. So for continuous assessment, what I do is for everyone, the base point I give to everyone is 10 marks, which is for attendance, because I assume that everybody attends lectures. And even if you didn't attend one or two, you still had opportunity to sit down and watch the video and engage with it. So 10 marks for everyone for attendance, which is the base. It means you are left with 30 marks for the continuous assessment, because continuous assessment are supposed to have 40 and exams should carry 60. So if you have 10 as the base for everyone, then you are left with 30. So what then happens to the 30? So you see, I talked about giving you the self-assessment questions, and your engagement with it is going to reflect in the exams. So the rest of the 30 will depend on your performance in the exams. What it means is that if you have 60 over 60 in exam, it means you have 30 over 30 for the rest of the 30 marks for the continuous assessment. If you have 20 in exams, it means for the rest of the continuous assessment, you have 10. It's just half of what you get, because you are doing 60 in exams and you are left with 30, which is half of that 60. So whatever you get in exams, half of it is what you have for the rest of the 30 marks for the continuous assessment, which is why it's serious. You have to be serious, responsible, sit down and do what you are supposed to do. I will try as much as possible, give you all the materials, every information you need to facilitate your studies. But in the end, the buck stays with you. You have to sit down and study. All right, so I think at this point, everything is clear about how the course will go, how we'll take it. Now, every time at lectures, all your microphones, all your cameras will be disabled. And when I finish, if you have any questions, you can raise your hand, or maybe even along the line, anyone, if you have any question. But usually it's better that the lecture flows, and if you have any questions, you write those questions down. Actually, the chat is working. I'm monitoring the chat as well. So if you write in the chat, I'll see it. So you can use the chat. If there is anything, you can write in the chat. I'll see it and respond to it. Alternatively, you can wait, and at the end of the lecture, once you raise your hand, I will enable your microphone. I'll call you, you speak, and then I will respond to you. That way, the class will be quiet throughout, no noise, and the videos too will be well recorded, so that you can watch them later and make meaning out of it. All right, so I think we are done with this introduction of the course to you, so we can dive into our main lecture for today, which is going to be on the naming and classification of enzymes. That's what we'll start with, the first topic. So we can move on to that one now. If you have any questions, like I said, can use the chat. You can write in the chat. I'll see it. If there is any issue, anything happening, you can write in the chat. I'll quickly attend to it. All right. Let's move on. Okay. As I mentioned earlier, today we are starting with enzyme nomenclature or naming of enzymes and classification. But even before then, we'll take some historical background to this. Somewhere getting to the end of the 18th century to the beginning of the 19th century, scientists have known that there has been the possibility, or let's say the digestion of food substances like proteins by extracts from plants. So if you do they use some extractions from plants, and if they add it to say things like meat or some carbohydrates like some sugars, they could see that there was some activity on these substances, but it wasn't understood what really was happening. Okay, so somebody says you cannot see my screen. Eugene Mensah. Perhaps it's coming from you, because as I monitor on my other device, I can see the screen. So maybe you should go out and come back. That's what some students do, and it works for them. So it's possible that you are having some network issues. That is why you are not seeing the screen. So you can go out and come back in, and I'm sure you'll be able to see the screen. In any case, the video is being recorded. So even if you have any issues later, you can take the link and watch it. So as I was saying, even though getting to the end of the 18th century, beginning of the 19th century, scientists have known that there is this digestive action of extracts of plants, for instance, on food substances like proteins and sugars. The mechanism by which this occurs wasn't that known until a French chemist, Enzyme discovered what is referred to as diastase, working with some colleagues in a sugar factory. They discovered the enzyme diastase that was extracted from malt solution. Of course, today, that if you say diastase, even though that's how it is called, that's how it was called diastase, but now diastase is what we are referring to as amylase. So it can be any of the amylases. It could be an alpha amylase, beta amylase, gamma amylase. All of these, the group of amylases is what was being referred to at the time as diastase. So they could see that there was some extract from malt that was having some process of digestion, or say it was actually fermentation which was occurring with these sugars. Now, some 1 or 2 decades later, another French chemist, he's regarded as a chemist at the same time, a microbiologist. You can say he was a biochemist because he was doing both chemistry and microbiology at the same time. Discovered that the sugar fermentation which was occurring was actually occurring in the living cells of yeast, which were being added to the production mixtures of carbohydrates, say in the production of BS, or say alcohol. Because up until then, what was being taught was that the fermentation process which was occurring was actually as a result of the dying and the decay of the yeast cells, as if it was the decaying of the yeast cells which was causing the kind of fermentation effect that was being observed. But Louis Pasteur came up with the discovery that actually the fermentation process was occurring in the living cell, and not that the cell was dead and decaying. That's why it was causing that fermentation. And he was the first to introduce, that term, the term ferments. So he gave the name ferments to the substances which are in the yeast, which are responsible for the fermentation process which was occurring, or the digestion process which was occurring on the sugars by yeast. Now, later in Germany, we had Wilhelm Kohn, is a German chemist, a German physiologist. He was the first to actually use the term enzyme, which he coined from the Greek word enzymon. Enzymon refers to the process of leavening that is in baking. So when you add yeast to the dough, and due to the respiration which is occurring in the yeast, it's releasing, say, carbon dioxide, which would then result into the dough to puff up. The bread becomes, bloated up, and so when it's baked, it can be filling. And so he referred to that the substances which are responsible for the digestion processes as enzyme. And since then, this term enzyme has been used for these substances that are responsible for the digestion processes which were being observed. In English literature, we have been referring to it as enzymes. Even though in some languages, some Slavic languages, like the Russian language, the maybe Kazakh, Uzbek, Turkmen, all these languages still use the word ferment to refer to the substances that are responsible for these digestion processes. But mostly in English language, even in those Slavic languages, sometimes you hear them translating the English literature, also calling these substances as enzyme, and not using the word ferment for these things. Then Edward Buchner, another German, who actually won a Nobel Prize for chemistry, for discovering what is referred to as cell-free fermentation, because at this point it was being thought that the fermentation process or the digestion process which was occurring was actually occurring only when there were living cells, the yeast cells, present inside these solutions with sugars in them. But Buchner discovered that even if you do not have the yeast cells in a mixture, if you have just an extract from the yeast cells, the same process occurs, which means that you didn't have to add the yeast itself, but if you have an extract from the yeast, which is a non-living thing, it's a non-living substance which you are adding there will still be that process of fermentation occurring. And so it wasn't that the process only occurs when there are living cells, because as you saw much earlier, Louis Pasteur here actually was referring to the fact that the fermentation process was occurring inside the living cells, and not that the cells were dead and they were then because of their decay that the fermentation was occurring. So from Louis Pasteur, it was thought that the process only occurs when there are living cells of yeast in a mixture. But here comes Buchner discovering that it is not only when the yeast cells are there living that you have the process occurring, but even if you have an extract without any living organism, not the yeast itself, in a mixture, you will still have the digestion process. So that was a new knowledge that was being added there in this case. Right. Now, later in the US, we had James Summer, John Northrop, Wendell Stanley. These three scientists got the Nobel Prize in chemistry in 1946 for their conclusive work, that found out that enzymes were proteins. But, before this, there had been disputes among scientists. Some scientists presume that, yes, enzymes were proteins. Others presume that no, enzymes were not proteins, but that the proteins were just carriers of the enzymes themselves. So what enzymes were actually was not still understood. What were these things which were causing these digestive processes? It was still not clear at that time. But the work of James Summer, which resulted into the isolation of the enzyme urease from jack beans in 1926, and then later also he was able to extract the enzyme catalase from beef in 1937. So at this point, once James Summer was able to isolate or crystallize these enzymes, it became now much more easier to study enzymes, the structure of the enzymes, and even to now study the mechanisms of these enzymes. Because of course, now once you are able to separate these substances, then you'll be able to study them. Before then, they were not able to separate them. If you are not able to separate a substance from, say, a sample, say from a living cell or from a living tissue, how are you going to, study it into detail? So James Summer did a good work there by isolating these enzymes. And once he was able to isolate these enzymes, it then resulted into the development of a procedure for isolation or crystallization of these enzymes. And so John Northrop and Wendell Stanley, studying these three enzymes, pepsin, trypsin, chymotrypsin, you will come to see that these enzymes are involved in the metabolism of proteins. And in their studies, they conclusively came to the point that, yes, enzymes were proteins. So that was a breakthrough there, that enzymes were proteins, because then they were able to, now once these enzymes were separated or isolated, they were now being able to be studied by X-ray crystallography to know the structure. And once you know the structure of these substances, you'll be able to now study the mechanisms of action of these substances, and much more information can now be known from these studies. And now it is now in modern times now, it is now very much known that, yes, enzymes are biological catalysts. So these are substances that increase the rate of biochemical reactions. And even though it was concluded that enzymes were proteins, it is now well known that, yes, most enzymes are proteins, but not all enzymes are proteins. You actually do have some ribonucleic acids, RNAs, which we refer to as ribozymes. These are also biological catalysts. These ribozymes, act in processes of metabolism of nucleic acids themselves. So you find them in the processing of nucleic acids when they are being synthesized. You have ribozymes that cut off some mistakes in the sequence of nucleotides in these nucleic acids in order for them to join back and have the nucleic acids having the right sequence of nucleotides that will make them functional. So apart from most enzymes being proteins, we now know that some enzymes are also RNAs. And you want to think of it, understand it that, what really is the importance of these enzymes? Why do we need enzymes in the first place? And you see, you have, let's take for instance that you have sugar in bottle that is in your kitchen or it's on your dining table. And the sugar is there for many days, months, years. If you don't touch it, the sugar will still be there. Maybe the only thing you will notice is because sugar is hygroscopic, it can absorb water from the atmosphere. If it is a small amount of sugar, maybe it will become watery. Or sometimes actually the sugar actually cakes because it absorbs water and becomes sort of bundled together. Maybe that's what you notice, but the sugar doesn't vanish. You don't see that the sugar vanishes. And why am I talking about the sugar is gone? Because even from junior secondary school or even from primary school, you have always been talking about the metabolism of carbohydrates, that carbohydrates break down into carbon dioxide and water and give energy. So when you talk about digestion, you've always talk of the fact that carbohydrates give energy, and during the digestion of carbohydrates, the byproducts that are produced is carbon dioxide and water. That's what you talk about. And yet, when you have sugar, so if the equation which you are seeing on your screen, this is sucrose, C12H22O11 sucrose with oxygen. So sucrose reacting with oxygen, producing carbon dioxide, water, and then there is some amount of energy which is released in that reaction. So when you talk about metabolism of carbohydrates, you're actually referring to this reaction. Of course, this is an oversimplified process of what really happens with carbohydrates, because as you go ahead in biochemistry, maybe you have already done this in some other course when you saw metabolism of carbohydrates, looking at the processes of glycolysis and the Krebs cycle, for instance. And so you have seen that there are many steps that are involved in these processes. But here we see this process being oversimplified here, that you have a sucrose, which is a disaccharide breaking down to give you carbon dioxide, water, and energy. And yet, when you put down the sugar in your kitchen, in your dining hall, the sugar does not turn into carbon dioxide, evaporates, water produced, and then the energy is given. You don't see that happening. Why is that? Why is it that we are saying that when you take in the sugar now turns into carbon dioxide, given out as a byproduct, some water is produced, and then it gives you energy? How does that occur? Now, what actually it means is that the reaction of the sugar to produce, carbon dioxide as a byproduct and water and give out energy, it's actually spontaneous. It is occurring, but that process occurs very, very slowly that it becomes negligible. You can't notice it that much. But the moment the sugar gets into, the living system, enzymes are catalysts. These are, we said from the beginning that these are biological catalysts. So biological catalysts, what's a catalyst? A catalyst is a substance that increases the rate of a chemical reaction. And you must know that a catalyst does not come to cause a reaction to occur. No, it only comes to increase the rate of a reaction which is already occurring spontaneously. So that if a reaction will not occur, and you put these reactants together and add a substance to it as a catalyst, that substance is not going to cause a reaction to occur. The reaction must already be occurring before the catalyst comes to increase the rate of it. So that's why I said, yes, this reaction is occurring all right if you put the sugar there, but because it's exposed to oxygen, but it is occurring at a very, very, very slow rate, so negligible that you will notice it. But once it goes into the living system, the enzymes increase the rate of that reaction so that energy is generated, which is really, really important because you come to realize that without enzymes, what it means is that these chemical reactions will be very slow and cannot sustain life. But life is supposed to be sustained, because all of these things, the activities we carry out as I speak, it's a lot of chemical reactions that are occurring in my muscles of the jaws, even my hearing as I speak. I listen to myself. My brain is processing what I'm saying, coordinating everything. If I make a gesture with my hand as I speak, all of these things are being, controlled by chemical reactions. Same way you listening and watching on the screen, you have chemical reactions that are occurring in your eyes, in your ears, and you can hear, you can see, you can understand. Your brain is working. All these things are being coordinated. So there has to be these chemical reactions that are occurring at a very, very fast rate and coordinated such that life will be sustained. So you can see how really, really important enzymes are as far as the sustenance of life is concerned. Now, okay, so I think, Taria is saying that do enzymes in living cell work at the same rate as extracted ones? All right, so what will happen is when we come to factors that affect enzyme activity, it will answer that question, whether the enzyme in living cells work at the same rate. If you actually provide the same conditions that are in a living cell in a lab environment for the enzymes, is going to work at the same rate as it works in the living cell. And those factors usually, we'll come to it, you'll see the factors: temperature, pH, concentration, all of these things work together to give you the rate at which the enzyme works. So you see that is why when you are doing experiments in the lab, you actually provide some condition for the enzymes uh, to catalyze a specific reaction. And most of the times it's about temperature and pH that you will be controlling. It's the same thing if it is in the industry, for instance, that enzymes are being used, maybe in the food industry, the same conditions. The enzyme is known to be active at a temperature of, say, maybe 30, 35 degrees Celsius. It is that temperature that is provided for the enzyme to be most active. And then a pH is also controlled in the system. So yes, it will work at the same rate if the same conditions are provided for it. All right, so I hope Tavia, you are okay with that. All right, so all catalysts, whether it is inorganic or organic, like so we're talking about enzymes which are in living cells, all catalysts, act with the same principle, what we refer to as the activation energy profile, which is to say that if you take this energy profile where on the vertical axis we have energy level, that's an energy level that reactants will be, and on the horizontal axis we just have some reaction coordinates there, which is to say that, so if you have this level somewhere in the middle here, you have reactants at a certain energy level. Now, what you need to understand is if you put two substances together, reactants, what is going to make those two substances react to give you a product? Now, I make an analogy all the time to explain this for students to understand, which is to say that, you see, at every point in time, even as human beings like this, that's at an organismic level, you always want to be in a comfortable position. So, for instance, as you listen to this lecture, as you are seated or you are lying down, whatever it in whatever position in which you are now, if you are comfortable in that position, you will be there in that position throughout. But if you feel a certain discomfort, you will move a little and to put yourself in a comfortable position. So at all times, you want to be in a comfortable position. Now, if you are comfortable at a certain position, there will be no point for you to move from that position to another position. So it's the same thing to do with, say, molecules. If you are talking about reactions, you are talking about the interaction between molecules of substances. Now, what is going to cause molecules of substances to change from the form in which they are into a different form? If those substances are at an energy level, which usually we refer to as a ground state, that they are stable. If they are stable, there will be no need for them to move from that energy state in which they are stable to form another substance. No, they won't do that. And so what it means is that the substances will have to be activated to go into an energy level in which they are unstable before they will then look for a new energy state, which will become more stable. And you must also know that when you refer to a substance, if you mention any chemical substance, that substance will not exist if that substance is not in a stable form. The substance exists because it is in a stable form. That is why it is there. If it is not in a stable form, it will change into a stable form into another thing. So same thing, you have reactants which are stable. If you take any substance, like maybe you are talking about, say, sugar and oxygen, these substances are stable substances. Now, what it means is that for these two substances to react, it requires what we refer to as an activation energy. So the substance will have to be activated, the reactants will have to be activated to a higher energy level, what we refer to as transition state. And then in that transition state, these two substances, being unstable, react, form new products, which are different substances, what we are referring to as products, which will then be having even a lower energy. So you see the product is at a lower energy level, a more stable energy level there. Now, understanding this principle, what then is the enzyme doing in this process? What is the enzyme doing? Now, if you take the energy profile that we are seeing on your screen, you have one energy profile which is in blue. That profile is for a non-catalyzed reaction. And you see another one which is in orange. That energy profile is for a catalyzed reaction. Because we are talking about enzymes, we are referring to the blue as a non-enzymatic reaction, and then the orange as an enzymatic reaction, which means it's being catalyzed by an enzyme. The reaction is being catalyzed by an enzyme. Now, what you notice is that the enzyme comes in to reduce the activation energy, which is required for the reactants to move to a transition state. So you see the transition state for the non-catalyzed reaction is high. More energy is needed to move these reactants to a transition state before they react to form the product. But for a catalyzed reaction, you see the catalyst first forms an enzyme substrate complex, which is has a little bit lower energy level than the reactants themselves. And that enzyme substrate complex requires lesser amount of activation energy to move to a transition state. And then you have that transition state now being converted into an enzyme product complex, which then dissociates and a product is formed. So the product is going to be the same for whether it's a non-enzymatic reaction or an enzymatic reaction, but for the enzymatic reaction, what is happening is that because you have the activation energy lowered, you have the reaction occurring faster. Lesser amount of energy is needed, lesser time is required for the reaction to proceed to produce the products or to form the products. All right, so that's a basic principle for all catalyzed and uncatalyzed reaction, which is the same for enzymatic and non-enzymatic reactions. Right. Now, before we even continue and dive in more and more, we're going to be making use of some terminologies that we should quickly get acquainted to, so that when we start to talk about those things and we mention those words, we will all be on the same page and understand. The first is cofactor. Now, a cofactor is a non-protein. You see, we've talked about the fact that enzymes are mostly proteins. but we do have some non-protein parts that are required to be attached to the proteins in order to activate these proteins to carry out their catalytic function. These we refer to as cofactors, and cofactors are usually metal ions, like iron, Fe2 plus, copper, Cu2 plus. You see these being referred to magnesium, Mg2 plus. You see them a lot of times acting as cofactors. These are inorganic, metal ions that are acting as, call them helpers or say activators of these proteins. Now, even though you see in biochemistry books, the term cofactor and coenzyme are used, interchangeably sometimes, but there's actually a distinction between these two. That usually when coenzymes are being referred to, they are also non-protein, but they are organic molecules. They are not metal ions. These are organic molecules. You will frequently see them as vitamin Bs. We'll come to see, these the different forms of vitamin Bs, and what you do refer to as vitamin B complex, for instance, containing different forms of vitamin Bs. You come to actually see why a lot of times when medicines are being prescribed are being given with vitamin B complexes, because these act as activators of enzymes, which will be catalyzing the metabolism of those medications that are being given to patients. Of course, if a person is sick, that person's system is having some impairment, and so if some medications are being given to the person, then there should be some boosting to the system to metabolize those medications in order for the intended activity or the intended function of the medication to be carried out. So we see that these different forms of coenzymes. Now the enzymes themselves, the proteins, you see them being referred to as apoenzyme. Sometimes you will see also being referred to as an apoprotein. You actually see this term not being used for only enzymes like the apoprotein. They can be used for other substances like hemoglobin, for instance. Because I think when we were doing proteins in first year, we did look at, say, the structure of hemoglobin. For instance, that hemoglobin contained the central ion, as a tetramer. You see that protein is also being referred to as an apoprotein, because you have it having a prosthetic part, which is ion, Fe2 plus, together with the protein before you have it being functional. Now, what is an active site? We're referring to enzymes as mostly proteins. Of course, you do have some RNAs also there acting. All of these are biomolecules. And you know that, if you say an enzyme is a protein, the protein can be large, consisting of lots of amino acids. It could be maybe oligopeptide, or it can be even larger polypeptides. But that large protein, which is the enzyme, is not that the whole surface of it, any point at all, can be interacting with reactants and be catalyzing the reactions of those molecules. But you have these enzymes, these proteins having some specific portions, particularly some amino acids, which are in the sequence of the protein structure, that they act like they form something like a pocket in which the reactants get into, and then reaction occurs at that particular point of these proteins. Those particular points are what we refer to as the active site. So you see this term also being used, the active site of an enzyme. So it's like a pocket, as a three-dimensional cavity of the enzyme with some specific amino acids that are going to allow for the substrates or the substances that are reacting to react at that particular portion. Of course, any process which initiates the activity of an enzyme is what is being referred to as activation, just from the word. It also means that any substance which is causing the activation of an enzyme is an activator. So, for instance, we did refer to, say, these cofactors, coenzymes. When they bind to the apoprotein, they will activate it. So they are activators. And the opposite of that is inhibition. So if the activity of an enzyme is being slowed down, or the enzyme is being made inactive altogether, that it does not act at all, that process is an inhibition of the enzyme. And whichever substance is causing that is what is being referred to as an inhibitor. Any compound that binds to the enzyme, either it lowers its activity or completely makes the enzyme inactive. We'll come to see how some of these processes of activation and inhibition are, explored in, say, drug discovery, for instance. Then, of course, the substances themselves that are reacting are what we refer to as the substrates. So those are the substances on which the enzyme is acting to accelerate their reaction. So these terms, we'll be making use of them as we speak. As we look through, when we talk, we'll be mentioning these words. We say substrate, then you know that we are talking about a substance on which the reaction is occurring. Inhibition, inhibitor, activation, activator, active sites, apoenzyme, cofactor, coenzyme, all of these terms now clear to you. So when you hear it being used, you will understand. We'll move on to look at naming of enzymes. Naming of enzymes. How are enzymes named? You can actually tell what an enzyme does just by looking at the name of the enzyme. So one of the forms in which enzymes are named is by adding the suffix ase, A-S-E, to the name of the substrate on which the enzyme is acting. So, for instance, the enzyme urease. Urease is the enzyme that catalyzes the breakdown of urea. So the substrate is urea. So you can see the name urea, of course, there's A there, and because of that, the A is not repeated. So just adding the A's, you have urease. So when you see the name urease, you know, yes, this enzyme acts on urea. It catalyzes the hydrolysis or the breakdown of urea. Lactase. Lactase catalyzes the metabolism of milk, sugar, lactose. So again, the substrate is lactose. and the enzyme is lactase. So you see the names of enzymes named based on the substrate on which the enzymes are acting. Another way that enzymes are named is by the activity or the reaction that the enzyme is catalyzing. So you do hear the name DNA polymerase. And what does DNA polymerase do? DNA polymerase catalyzes the process of polymerization of nucleotides. So the process is polymerization. That is bringing nucleotides together, binding the nucleotides together to form a polymer. And that polymer is the DNA. The deoxyribonucleic acid is being formed from nucleotides. And so the process is polymerization, and the name coming from that process polymerization is polymerase. All right, so Jesse Oyote says, please, does it mean if a person is lactose intolerant, can he be given the enzyme lactase to speed up digestion of lactose? All right, so what will happen is we'll actually come to things like this, like the lactose intolerance, what actually it is, and all of that. So you hold your gun. We'll come to all of these things, and you'll see. That is to talk about maybe some deficiencies and how to, uh, deal with some of these things. So we'll come to it. So don't worry. All right. So, but of course, the idea you have, yes, of course, that's what it is. That if a person is lactose intolerant, if the person is, if some analog of lactase is given to such a person, it means that lactose is going to be broken down more freely. But of course, the problem would have already occurred, because what happens is that someone who is lactose intolerant will not be producing it's not having this the enzyme lactase being produced in adequate amounts. And so what happens is, in the end, you have the gut bacteria which will be acting on lactose and be producing gas, which will be causing bloating in such a person. And so the sugar lactose had already gone into the large intestine, and bacteria is already acting on it and producing such. Of course, yes, if you introduce a lactase analog, of course, it will go to manage the problem. So yes, your thought is accurate. All right. So we do see pyruvate decarboxylase. What is pyruvate decarboxylase? Here again, we see ase at the end of decarboxy. And that's because the process which is happening with pyruvate decarboxylase is the removal of carboxyl group from pyruvate. So as I was saying, I hope you did say metabolism of carbohydrates, glycolysis, Krebs cycle. Did you do that in first year? Maybe last semester. I don't know. Maybe you did it. If you did it, then you would have come across in a process of glycolysis, you came across pyruvate. and that pyruvate undergoes decarboxylation process and before going into the Krebs cycle. So I hope you did that. So, but of course, see, this is just to tell you that once you see the name carboxylase, again, you see ACE. ACE is being added to the process. So this time around, it's not the substrate, but the process. And so you see pyruvate, decarboxylase means there's a removal of carboxyl group from pyruvate. That's why you have the name of the enzyme, that way. Lysozyme, which you also see acting on bacterial walls. Actually, based on these, you have some antibiotics being produced. We'll come to see some of those things causing, the breakdown of the bacterial walls. That breakdown, the process of the breakdown of the bacterial walls, is what you refer to as lysis. Because it's a lytic process. And from it, you have the name lysozyme. This time around, it's not ACE. It's not the suffix ACE, which is being added, but you have a suffix zyme that is being added there. Now, coming from the name enzyme, you have that lysozyme, being added there. Of course, I think looking when we were talking about the historical background, I think one of these, I think, was it Buchner actually named the enzyme, which he found to be acting on sugar, as zymase, for instance, in the fermentation process. We refer to it as zymase. All right. Apart from these processes of the system of naming which we just looked at, there are also common names which don't follow any of these systems that we refer to, adding the suffix "ase", or say you see zyme being added, at the end of activity of the enzyme. You don't see that. For instance, for the enzyme pepsin, a common enzyme, found in the digestion and breakdown of proteins. It's one of the first, as we looked at the history, saw that it was actually one of the first enzymes that were isolated. You saw pepsin. Pepsin, just comes from the Greek word pepsis, which means digestion, because it was first attributed to, the digestion of foods, and so the name pepsin was given to it. It doesn't follow any of the naming systems that we talked about earlier. Trypsin, another common enzyme known for the digestion of proteins, is also a common name. It's also coming from the Greek word trying, which means to wear down, and it's because of the process by which the enzyme was first isolated. It was first isolated just by rubbing pancreatic tissue with glycerin. So so glycerin as a solvent, which was used to just rub the pancreatic tissue of a laboratory animal, and then you have the enzyme trypsin being secreted into glycerin. And because of that process of wearing down, you had the name trypsin being given to that enzyme. Of course, this was before you then have this system of naming enzymes introduced. Now we can move on and take a look at classification of enzymes. classification of enzymes. Generally, we have 6 main groups of enzymes, which are based on the type of reactions that are catalyzed. And the first of these passes is what we refer to as oxidoreductases. Now, from the name oxidoreductases, what it means is that this group of enzymes catalyze oxidation and reduction reactions. What you usually refer to as redox reactions. You do have a lot of redox reactions occurring in biochemical systems. And these reactions are catalyzed by oxidoreductases. Sometimes oxidoreductase they are also referred to as dehydrogenases. So you see, an example, lactate dehydrogenase. And what is lactate dehydrogenase doing here? So that is lactate or lactic acid. It's a product as a reactant, which is being converted to the product pyruvate. And what is really happening? Now, when you look at the structure of lactates and pyruvate, what's the difference? The difference you see is on this carbon in the middle. There are 3 carbons here. You have a CH3, CHOH, and then you have COO. So that's a carboxylic group or carboxylic acid group there. And if you look at these two, lactate and pyruvate, what's the difference? The difference you notice is that there is a hydroxyl group OH on the central carbon there. In place of that OH, you see there's a C double bond O there. So what is happening? The process of oxidation. So lactic acid oxidized to give you pyruvate. And the enzyme which is catalyzing that reaction is lactate dehydrogenase. So that's an oxidation reduction reaction occurring there. All right. Transferase. That's the second class of enzymes, transferase. And what do transferases do? Again, from the name, they transfer functional groups between molecules. From one molecule to the other, these transferases transfer functional groups. And we're seeing an example here, aspartate aminotransferase, or sometimes being referred to as aspartate transaminase. From the name, it tells you that there is a transfer of an amine group. Transfer of an amine group. Now take a look at this. If you look at aspartate or aspartic acid, there's an amine group on that carbon there. And if you take alpha ketoglutarate, you see there's a C double bond O also on that carbon there. Now, in place of this C double bond O, you see there is an amine group in the product glutamate. And where is the amine group coming from? You see the amine group here on aspartic acid. In place of it, you have a C double bond O in the product oxaloacetate. And so what is really happening is an exchange between this amine group in aspartic acid and the double bond O here you have in alpha ketoglutarate. And so you do have that exchange between these two groups, and you have oxaloacetate. In place of this amine group, you have a C double bond O, and a glutamate, you now have an amine group there. You actually see this process in the metabolism of proteins, what we refer to as transamination. You see this process occurring in there. So that's a transfer of functional groups as the second class of enzymes. We have a third class of enzymes being referred to as hydrolases. These catalyze the process of hydrolysis, hydrolytic reactions, which is a breakdown of a molecule, in the presence of water. And we see an example here, acetylcholine esterase, which is breaking down acetylcholine. In the presence of water, you have that, this acetic group part of the molecule broken, and you have an acetic acid, and then the choline portion is also separate. So water acting in that process, of course, you have H plus OH, and you have the H plus going to the acetic acid there to give you the full molecule acetic acid, and then the OH goes there for the choline. So that's a breakdown. It's a hydrolytic reaction, a breakdown, hydrolysis of acetylcholine into acetic acid and choline. So this is also acetylcholine esterase. Yes, we'll see some application of acetylcholine esterase with some analog succinyl choline, which is used to slow down, as a muscle relaxant in minor surgeries. We'll take a look at something when we are looking at some practical applications of the knowledge. We'll come to see things like that. All right. Now, the fourth group, we said we have 6 common groups, so the classes. So we have the fourth, which is referred to as lyases. And what do lyases do? Lyases, either they will remove some groups from a molecule, which will result into the formation of double bonds, or they will add, which is either they remove or they add. If they are adding groups, then double bonds will be removed. So for instance, we see the 6 aconitrates. If you did a Krebs cycle, you would have seen this kind of reaction there. And you see there's a double bond here between these two carbons. There's a double bond there. 6 aconitrates. Now with the addition of water, what do you notice? You notice on this carbon, there is an OH. And on this carbon, which was carrying the carboxylic acid group, a hydrogen has come to add, and the double bond is removed. There can be the reverse of this reaction, where you have water removed from isocitrates to give you 6 aconitrates. So it's either a group is being added to remove a double bond, or a group is being removed to form double bonds. And this class of enzymes we refer to as lyases. The fifth, the name will even tell you, isomerases. So isomerases, transfer groups between molecules to form their isomers. So for instance, you have the enzyme phosphohexose isomerase, which is converting glucose 6-phosphate to fructose 6-phosphate. So what are isomers? These are compounds that they have the same chemical formula, which means they contain the same number of atoms of elements, but they have different structures. So these are isomers. So glucose 6-phosphate and fructose 6-phosphate are isomers. It's just a rearrangement of the atoms of the molecule glucose 6-phosphate will give you the structure, the fructose 6-phosphate. So from a pyranose structure, you have now a furanose structure. And the enzyme involved in that, phosphohexose isomerase. That's an isomerase. Right. Again, if you did glycolysis, Krebs cycle, you would have come across some of those isomerases in those processes. And then the last group here is ligases, sometimes also being referred to as synthetases, which are involved in the joining of molecules, 2 more molecules to form a bigger molecule. These we refer to as ligases. Now, even though we said 6 classes of enzymes from the beginning, and we have exhausted that up to ligases. When you read biochemistry books, you may also come across a seventh class of enzymes, which are being referred to as translocases. And I mentioned this particularly because the second part of the course is on biomembranes. And so if you take a look at the translocases, they are not actually being involved in catalyzing biochemical reactions, but these are transmembrane proteins which facilitate the movement of substances across cell membranes. But sometimes when the classification of enzymes are being talked about, they are also referred to as a seventh class, translocases. So you may see that in some books as well, translocases. But of course, those are not really involved in biochemical reactions, but rather just transportation of substances across biomembranes, except that maybe in some cases you have some activation processes, what we do refer to as some maybe channel gates, things like that, but they will be stimulated by some substance to open for the substances to go across them. But still, it is not a catalysis of a biochemical reaction there. All right. Now, based on specificity, how specific an enzyme is acting, you can also have enzymes being classified into 4 main groups. And these are absolute specific enzymes, which means that these enzymes act on only one and only one single substrate. They act on a single substrate. Nothing else, just one. Group specific will be acting on similar molecules, but they will have the same functional groups. And we'll take a look at examples of these for us to have a better understanding of it. You have some enzymes also which are bond specific. They act on some specific bonds or linkages in compounds. And then you have what we refer to as stereochemically specific enzymes. And these will be, you have enzymes which will be acting on either the D isomer of a substrate or the L isomer of a substrate. So this, should take you back when we were doing biochemistry in first year. I remember we looked at the structure of carbohydrates, and we did see D and L isomers of these. I think even when we were talking of proteins, particularly talking about the amino acids, we also saw D and L isomers. So here again, so you see, we are building up on the knowledge from first year. So nothing is thrown away. You will keep on building on it. So if you have thrown your notes away, I'm sorry. You must always have those things, because these things will be repeating from first year all the way to the last year. So these four groups, let's first take a look at an example of absolute specific. So we did talk about the enzyme lactase. That enzyme acts only on lactose. Can you imagine if this enzyme was acting on a different substance apart from lactose, and we give it the name lactase? That's going to be misleading, because then the other substance on which the enzyme will be acting is not considered in naming the enzyme. But it is because you have this enzyme lactase acting only on lactose. That is why it's possible to name it lactase. And so it is absolutely specific. It acts only on, one substrate, lactose. And so you see the reaction being shown there. You have, so this is lactose. Lactose is disaccharide from two molecules of galactose, which are bound together to have okay, so somebody says I should speak a bit louder. I'm actually speaking loud. I think, sometimes maybe when the network is falling, then maybe the voice goes down a bit and then maybe it can come up. So I'm sure it will come. All right, so you have lactose, which is a disaccharide from um, uh, we call it galactose and glucose, with that glycosidic bond between them. And we see the enzyme lactase, it will be acting in this process, breaking down. So you have this glucose molecule, which is broken off, and then you have the galactose molecule there. So what you actually see here is that we are seeing some intermediates where you have galactosyl intermediates. What actually it means is that the lactase, so this is a lactase molecule there. Well, it's a protein. That's the C terminus, the carboxylic acid group end of that protein. And you see the R, meaning that's the rest of the chain. And what you actually see here, it means that the active center of lactase, there you have glutamate, the amino acid glutamate. It's a big protein, and so that amino acid is at a position of 1749th position if you take the amino acid sequence in that enzyme, that protein. This glutamate, this amino acid glutamate, is what is having some we call it a nucleophilic attack there on that glycosidic bond and breaking it, so that you have this now lactase, which is binding to the galactose molecule to give you that intermediate, the galactosyl intermediate, which later is hydrolyzed. So you see water hydrolyzing the enzyme off from the galactose, and so the galactose molecule is free, the glucose molecule is removed there. So that's lactase. Of course, this is just to show the mechanism of action of this. As we move on, at least we should be looking at some mechanisms right from now. Even though there will be time we'll look more and more at mechanisms of action. So that's an absolute specific enzyme, lactase, acting only on lactose. Another one is glucokinase. Glucokinase. So again, from the name, glucokinase will act only on glucose. Now there's a new thing here that you should keep in mind. Kinase. What's a kinase? Kinases are enzymes that are involved in the transfer of phosphate group from one molecule to the other. And where is the phosphate group being transferred here? The phosphate group is being transferred from ATP, adenosine triphosphate, onto the glucose molecule. And so you see on this carbon, now you have OH. Actually, you do have a CH2OH here. In place of that, you now have a phosphate group. So one phosphate group from ATP comes onto glucose to give you glucose 6-phosphate. And so then you are left with ADP, adenosine diphosphate. Once a phosphate group is transferred from it, it's left with a diphosphate. And the enzyme involved in that process is glucokinase. That's also an absolutely specific enzyme. That is why we are able to name it glucokinase, acting only on glucose. But we do also have a group-specific enzyme, which will be doing similar thing like glucokinase, but it's not going to be acting only on glucose, but will be acting on all hexoses, all 6 carbon sugars. And this is a hexokinase. We refer to these group-specific enzymes as hexokinase. So you see the similar reaction, the same reaction actually, glucose, forming glucose 6-phosphate. Hexokinase will also act in the same reaction. And the hexokinase can do the same for other hexoses like fructose, like galactose. Hexokinase will act on these 6 carbon sugars. And it is actually based on the knowledge of these enzymes, the glucokinase and hexokinase, that you have some systems of fasting blood sugar being developed. Now, in normal circumstances, you have hexokinase is at lower concentration in the system. Is constantly acting on 6 carbon sugars, on hexoses, but specifically after taking in food, you do have the level of glucokinase increasing to act specifically on glucose. That is to control the amount of glucose that is being released in blood, so that once you have more carbohydrates being taken into the system, glucokinase will be acting in converting this to glucose 6-phosphate. And of course, that phosphorylation reaction is an activation process, which then takes the glucose 6-phosphate into the glycolytic pathway to generate energy, and also further go into the process of the Krebs cycle. And so what it means is that if at a certain point in time the activity of glucokinase is high, let's say without eating you still have glucokinase activity being high, that will be a sign of some perhaps some diabetic condition, because the fasting blood sugar is not supposed to be at such high level, which will cause glucokinase activity to be high. All right. Now, I will say that we did proteins and amino acids uh, in first year. So you must revise that. You must revise the amino acids in order to understand certain activities of enzymes when we talk about them. So I think the next thing we talk about is, we're talking about specificity, and I've brought this to remind you that you need to go and revise this, because then we start to talk about some types of bonds which are between some specific group of amino acids. So if we say amino acids that are having aromatic side chains, you do remember these are phenylalanine, tyrosine, tryptophan. Now, we did talk about an enzyme like pepsin. Pepsin. Now, pepsin is group specific. Group specific in what sense? Now, it is group specific in the sense that pepsin acts on proteins that have aromatic uh, some aromatic amino acids, the bonds that they form with the amino acids that are adjacent them. You have pepsin acting in those proteins to break down those proteins, which means that if you take this amino acid here, which is phenylalanine. So this is phenylalanine. The side chain is um, this aromatic chain, the aromatic ring, which you see there. There's a carboxylic acid group, the C terminus of it, and you have the N terminus, the amine group end of it. And so you have pepsin will be acting on the peptide bond, which is formed between, say, lysine and phenylalanine, and the same between phenylalanine and threonine. So that is, specific to uh, the amino acids with the aromatic, side chains. It's group specific. We do have trypsin also being group specific, which will be acting also on the peptide bonds that are formed between the carboxylic acid group of amino acids, arginine and lysine, with other amino acids. And what is the common, what's the similarity between arginine and lysine? The similarity you see is the fact that in the side chain of um, arginine, you see there is an extra amine group there. In that, what we refer to as a guanidino group in arginine. And you also see, an extra amine group here again, also in lysine. Now, the bonds that these amino acids, arginine and lysine, will form at their carboxylic acid end, those can be broken down by trypsin. So that's also another form of group specificity. And then we have, see, you also want to remind yourself of, we did carbohydrates and we talk about alpha and beta configurations, where we said on the anomeric carbon, if you have the hydroxyl group pointing downwards, you have an alpha configuration of the monosaccharide. If you have the hydroxyl group pointing upwards, you have a beta configuration there. You do have enzymes, which is, say, amylase. You have an alpha amylase, which we have, human beings. We have the alpha amylase. I think I talked about this in first year, that our alpha amylase can break down only the alpha 1,4 glycosidic bonds between monosaccharides in polysaccharides, say in starch. But the alpha amylase is not able to break down the beta 1,4-glycosidic bond. And I think I did even mention to you an example of, say, termites having some symbiotic relationship with the protozoa, Trichonympha, which has the enzyme cellulase. And cellulase is able to break down the beta 1,4-glycosidic bond, which is found in cellulose, which is why you see that termites will be chewing wood. And when they chew the wood, actually it is that protozoa, the Trichonympha, that actually goes to break down the cellulose, into glucose molecules. And so that's a bond specificity. So alpha amylase is having such a bond specificity to only the alpha 1,4-glycosidic bond, but not to beta 1,4-glycosidic bond. And then we did also talk about L isomers, D isomers. So if we take, say, an L-alanine, D-alanine, you see that you have some enzymes, what we refer to as stereochemically specific enzymes. You will have L-amino acid oxidase, which will be acting. If you take two amino acids, you will see that the L-amino acid oxidase will be catalyzing the oxidation of only the L isomer of the amino acid. You also have the D amino acid oxidases, which will be acting only on the D isomer of the amino acid. So these are stereochemically specific enzymes. So you have the L amino acid oxidase, D amino acid oxidase. So this is what we refer to as stereochemically specific enzymes. All right, I think this brings us to the end of our lecture today. So if you have any questions, if you have any comments, any contributions, you can write in the chat. You can raise your hand. I will call your name, unmute your microphone, and you can speak. So at this point, I've been talking for all this while. I'll keep quiet and wait to hear from you. All right, so Owusu says the YouTube channel. Well, actually, my YouTube channel is not all that updated. That is because I currently don't have access to it. When I come to Ghana, I'll be able to work on it more. But I have another channel. I think let me just look for let me get a link and send it to you. It's the Russian version of YouTube. So that when you go there, I actually once I start giving you lectures, I'll actually have your own playlist so that you can go straight to that. When you get to my channel, once you go to your playlist, it's going to be only my things there. All right, you also I will send a link later, okay? Because I think I've been logged out. If I want to log in now, it's going to disrupt the lecture and all of that. So I'll just send it to the class rep later, and then she can share it with you. Or even when I upload the video and all of that, and I send the link, you definitely have the link. So that will be all right. So also, I hope that's okay. All right, so I'm quiet now. We have 6 minutes more for the lecture to end. I think yes, 6 minutes more. So if you have anything, you can raise your hand. I will enable your microphone, and then I call your name, you speak. Alternatively, you can write in the chat so I respond.